Hello, my name is Sam Raney, and I appreciate the kind introduction. It has been my privilege over the last several years to be able to lead the Office of Generic Drugs GDUFA Science Research Program as we've sought to bridge the knowledge gap that existed at the time and to be able to develop science and technology that would lead to alternative bioequivalence approaches, new ways, more efficient ways by which to develop generic drug products, specifically for topical dermatological dosage forms, in the hopes that ultimately this would enhance patient access to these important medicines. And today we have the opportunity to look at some of the future directions and emerging technologies of today, as well as to discuss some of the current thinking about the alternative bioequivalence approaches that are currently in an advanced stage of development and already in practical use. I'll disclaim here that the presentation reflects my own views and should not be construed to necessarily represent the FDA's policies. So with that said, let's begin by covering a few major concepts with Q1 and Q2 as well as Q3. So the components or the ingredients of a formulation, which we call the Q1 properties of a drug product, as well as the quantitative composition, the percent of each of those ingredients, or the percent of each of those, the percent relative amount of each of those components, or the Q2 characteristics of that formulation. These Q1 and Q2 aspects of a topical product, as well as how it's manufactured, modulate its physical and structural arrangement of matter. And this arrangement of matter is what we refer to as Q3. Q3 characteristics refer to those arrangement of matter. And this covers, its rheological properties, its pH, whether or not it has globules, these kinds of things that describe the structural arrangement of matter and its physical states. These Q3 characteristics influence the molecular interactions that essentially control the rate and extent of topical bioavailability. And it is this rate and extent to which the drug becomes available at the site of action or sites of action that are at the heart of understanding whether two products are going to be bioequivalent to each other. So one approach to developing generic topical products is to characterize the complexity of the reference product in terms of these Q1, Q2, and Q3 characteristics, in terms of the components, the composition, and the arrangement, the physical and structural arrangement of matter, and then to match the Q1 and Q2 and Q3 characteristics of the reference product. In other words, that the prospective generic product should match the reference product as closely as batch to batch of the reference product matches itself or is identical to itself. But there can be variation even among batch to batch of the reference product. So this brings up an important question. How closely should test and reference products be matched? To answer this question, we're going to cover today some of the data, a very small set of a large amount of data that we've accumulated by studying numerous test and reference products as positive and negative controls for bioequivalence to see how closely matched these test and reference products actually are for products that are bioequivalent. Here we have a series of metronidazole products. We have a cream and a gel, or rather a set of creams and gels, where the reference product is shown as well as an approved generic product, and then again the reference gel and approved generic gels. And here we have various Q3 characteristics for the two creams as well as the three gels. And what may be useful to look at is really, while we can look at a specific number like the work of adhesion and say, well, this is around 60 and these are around 40, so similar to each other for the gels, similar to each other for the creams, but distinctly different. It's often more useful to look at the data in its profiles of these larger ranges and contexts. And so, for example, we see here the rheological plots of the two creams as being very similar to each other and well differentiated from those of the three gels, which are very similar to each other and well differentiated also from another lotion that's not shown in this table. Similarly, when we look at the drying rate, we can see that the drying rate curves are very similar for the 2 creams here and for the 3 gels here. The drying rate is important because as the composition of the formulation on the skin dries over time, its relative amount of the different ingredients changes as some of those ingredients volatilize. And what also ends up happening is that the solubility of the drug changes because the vehicle is changing and it's drying out. And the thermodynamic activity as the drug becomes more concentrated and its solubility approaches saturation solubility, the thermodynamic activity changes, and this really helps to modulate the rate and extent to which it becomes permeating into the skin. And so these drying rates are important. As we can see here, it's very closely matched for the gels. And if we look at the time that it takes for 30% of the formulation to dry, or to have 70% remaining, so that would be about here, that's very similar for the 3 gels, but it's not exactly the same. Numerically, if you look at it, 5.5, 4.7, 6.5, but you see that this is very, very similar. And likewise, it's similar for the 2 creams, although numerically it may not be exactly the same. Now, are these products similar enough to be bioequivalent? Well, we have clinical evidence to demonstrate that these products are bioequivalent, both for the creams and for the gels. And some have suggested, well, could it be that because the time that it takes for the skin to heal, in other words, these clinical endpoints, because that takes several weeks, that is it that the skin is and the dermatological conditions are not that sensitive, that there could be differences in the rate and extent to which it becomes available in the skin? In other words, could there be differences in the pharmacokinetics of these products that come around from these small differences and you just don't pick them up clinically? Well, to answer that question, we looked at the in vitro permeation of these products through human skin, and you see that the rate and extent to which they become bioavailable in the skin for the creams and for the gels is very, very well matched. So we not only have clinical evidence of therapeutic equivalence or bioequivalence, but also pharmacokinetic data showing that products that are relatively closely matched in terms of their Q3 characteristics exhibit comparable pharmacokinetics, and the same is true here for the gels as well. And this is not isolated. This is 5 different products of different dosage forms of metronidazole, and we have very similar data for other drugs as well. So one important thing to understand here is that clinical evidence has demonstrated the bioequivalence of several topical generics that are not necessarily Q1, Q2, or Q3 the same as the reference product. And in fact, that an expanding body of evidence has demonstrated that these topical generics exhibit comparable cutaneous pharmacokinetics, not only comparable clinical efficacy. The question then arises, if these differences are still okay in terms of the product being bioequivalent, both based upon pharmacokinetics and clinical efficacy, at what point are you not similar enough, that you've deviated so much that now the differences are impacting the bioequivalence of topical products? Or in other words, when do Q1, Q2, or Q3 differences actually impact the bioequivalence of topical products, and what may be acceptable differences between a test and a reference formulation? Well, it helps to think a little bit about the Code of Federal Regulations when we think about this question, and to really focus in on the regulations as they relate to waivers of in vivo evidence of bioequivalence. So if you look at the CFR, the Code of Federal Regulations, it discusses parenteral solutions for injection or ophthalmic or otic solutions, and describes that these products that would be eligible for a waiver should contain the same active and inactive ingredients in the same concentration as the reference product. Really, this is a concept of Q1 and Q2 sameness. However, for topical solutions or solution-based foam aerosols, these products that would qualify for a waiver should contain no inactive ingredient or other change in formulation that may significantly affect systemic or local availability. So this is not necessarily Q1 and Q2 sameness. Topical products are not required to be Q1 or Q2 the same, but really what's relevant here is that whatever small differences may exist should be those such that they don't significantly affect systemic or local bioavailability. And this is a very helpful concept as we think about a framework for the underlying Q1, Q2, and Q3. And this is an evolving concept for topical dermatological products that began as we thought about the underlying components. If the underlying components of the test and reference product were the same, and you had Q1 sameness, and you not only had the same components but also the same composition, and therefore also had Q2 sameness, and you had the same arrangement of matter, the same physical and structural properties of the same underlying matter, then you would have Q3 sameness. And this is now evolving further into a concept of Q3 similarity, where you would have similar components and composition compared to the reference product, and similar physical and structural properties, where you may not exactly have the same Q1 and Q2 sameness, but any differences would qualify as being a difference that did not significantly affect local or systemic bioavailability. In other words, that there should be no difference that may significantly affect local or systemic bioavailability, which may be Q1, Q2 sameness, but not necessarily. So you might have small differences here, but acceptable differences because they don't affect local or systemic bioavailability. Or you may in fact have exactly Q1, Q2 sameness, but small differences like what we saw with the drying rate for metronidazole creams and gels, that one might say, well, it may not be something we can call Q3 the same, but it's very similar. Either way, we see that we have this concept of Q3 similarity. And when we think about the data, the evidence that we've seen of what it takes when you characterize these test and reference products, what it takes to ultimately have the same rate and extent to the site of action and to be bioequivalent, perhaps the question becomes, is the right concept here that the products should be Q3 similar, because is it the Q3 similarity that is really the right way to control it? What we see is that all of these products have something in common in that sense, that they are not necessarily Q1 and Q2 the same. However, there's no significant impact on bioavailability. So this evolving concept for topical dermatological drug products of Q3 similarity is one that I want to take a little bit of time to talk about, because while evidence certainly demonstrates the relevance of the concept of Q3 similarity, and it's also more practical to think about Q3 similarity, it's really important to be clear, what do these concepts mean in a practical sense? And in particular, at what point are you close enough that you're similar, and at what point are you no longer close enough that you are now different? In other words, what is the boundary that separates Q3 similar from Q3 different? And a related question is, what is the distinction between the Q1, Q2 sameness standard and the no difference standard that we've talked about, where it would be no difference that significantly affects local or systemic bioavailability? I want to take a few minutes just to hone in on these pieces. So we'll begin first with the concept of when we receive a communication from industry, because we have a product-specific guidance that recommends this no difference standard, that a formulation, the test formulation, the prospective generic formulation, should qualify under the no difference standard in order to be eligible for a characterization-based approach, to be suitable for that. So when we assess these formulations, what do we do? Well, it's important to understand that determining the suitability of these proposed test product formulations, their suitability to demonstrate bioequivalence by a characterization-based approach, an assessment of no difference in formulation is essentially based on the same principles as assessing Q1 and Q2 sameness, including the same tolerances of plus or minus 5%. So we're not talking about something where anything goes. That's not the case. It's not as if you can have any underlying components and that you can deviate from plus or minus 5% and everything is acceptable. That anything goes is not what this standard represents. It's essentially a Q1, Q2 assessment. However, An assessment of no difference for topical dermatological products evaluates whether certain components and compositions may be acceptable for a proposed generic product. In other words, whether certain differences relative to what would precisely be Q1 and Q2 may be acceptable. And how do we determine what's acceptable? Well, either based upon information available to the agency or based upon evidence submitted in an abbreviated new drug application or in an ANDA. In other words, based upon evidence in the ANDA that there is no difference between the test and the reference products in either the local or systemic availability of the active ingredient. So this might be evidence from an IV-PT study showing cutaneous pharmacokinetics, as well as evidence from an in vivo plasma PK study showing no difference in systemic availability, as well as other evidence. And this gives us a modular framework for a characterization-based BE approach that's based upon Q1 and Q2 sameness, or no difference anyway, and physical and structural Q3 sameness, or really similarity, as well as what's frequently included in terms of a drug release rate test, an IVRT, and often for emulsions or for petrolatum-based ointments. We also include an in vitro permeation test, and there may be other types of evidence that also support a demonstration of BE, like if there's an in vivo kind of a systemic site or mechanism of action, then in vivo pharmacokinetic studies may be one of the additional modules that's included among this evidence. For corticosteroids, vasoconstrictor studies may be utilized. For most products, a comparative clinical endpoint study is still a viable way to approach things. And often in silico quantitative methods can complement some of these other approaches. So in conclusion, generic topical products are not required to be Q1 or Q2 the same compared to the reference product. Generic products for topical products should contain no difference in an active ingredients or other aspects of the formulation that may significantly affect local or systemic bioavailability. In other words, generic products should have the same local and systemic bioavailability of the drug product. Certain components and compositions may be acceptable for a proposed generic product based either upon information available to the agency or based upon evidence submitted in an ANDA. And just to kind of come back to why create a concept of Q3 similar if it is so close to what is essentially Q3 sameness? Well, consider that when the Q3 attributes of such a product, the kind of product that we're talking about over here, the no difference that meets the no difference standard, when the Q3 attributes of such a product match those of the reference product, but the underlying matter is technically not exactly the same, then one might not say that you've got the same arrangement of matter because the underlying matter may be slightly different. So it would not be considered Q3 the same, but rather Q3 similar. So both from a technical perspective as well as from a practical perspective, and based upon the evidence that we're seeing, the concept of Q3 similar may be the right way to think about these products. But from a practical perspective, when you are developing these products or when we're assessing these products, it's important to understand that this is still essentially based upon Q1, Q2, and Q3 sameness, with allowances made really based upon evidence of what is either information available to the agency to demonstrate that there's no difference for that component or composition, or based upon evidence submitted in the ANDA. So allow me to offer you the opportunity now to answer a challenge question. Differences in inactive ingredients or in other aspects of a proposed test product formulation relative to the reference products, these differences may be acceptable when? Is it that differences are never acceptable under any circumstances? Or is it that differences are fine, anything goes for topical products? Or is it that these differences may be acceptable when information available to the agency indicates that certain components and compositions of a proposed formulation may be acceptable, or that evidence submitted in an ANDA demonstrates that specific differences between a test and reference product do not alter local or systemic bioavailability, or C and D? I'll let you consider that for a moment. And the answer is C and D. With that, let me say thank you very much It has been a pleasure to have the opportunity to convey these messages that I think are quite important in terms of clarifying our current thinking as it relates to Q1, Q2, and Q3 similarity. There were a great many people involved in this, and certainly I'd like to thank others at the U.S. Food and Drug Administration, as well as our research collaborators. With that, I'll say thank you so much.